One of the most remarkable features of the immune system is its ability to recognize and respond to an almost limitless variety of foreign antigens, while at the same time remembering past encounters. How is this such precision and memory possible? The explanation lies in the clonal selection theory, a central concept in immunology that describes how specific B and T lymphocytes are activated, proliferated, and differentiated in response to an antigen. This theory was proposed in 1950s by Frank, and it remains the foundation of our understanding of adaptive immunity. The clonal selection theory is a cornerstone of adaptive immunology, providing a mechanistic explanation for how the immune system is able to mount highly specific and long lasting responses against foreign antigens while remaining tolerant to self. According to this theory, lymphocytes, B cells, and T cells are generated in primary lymphoid organs, with B cell maturing in the bone marrow and the T cell in the thymus. During development, these immature lymphocytes undergo genetic recombination of their receptor genes, producing a unique antigen receptor on the surface of each cell. B cell express B cell receptors that can directly recognize and bind to antigenic determinants, whereas T cells express T cell receptors that recognize antigenic peptides presented on major histocompatibility complex, which is MHC molecules. Importantly, each lymphocyte is pre-programmed to recognize only a single antigen epitope, which ensures that even before encountering pathogens, the immune system harbors an extraordinarily diverse repertoire of lymphocytes capable of detecting virtually any foreign molecule. When an antigen enters the body, it selectively binds to the lymphocytes carrying a receptor that is complementary to its structure. This selective binding is the critical clonal selection event. The engaged lymphocytes become activated and undergoes clonal expansion, a process in which it proliferates extensively to produce a large number of population of genetically identical daughter cells, all sharing the same antigen specificity as the original parent cell. This amplification ensures that there are sufficient cells to mount an effective immune response. Following expansion, the clonal differentiation into effector cells, which mediate immediately defense, and memory cells, which provide long term immunological memory. In B cell, effector plasma cells secrete large quantities of antigenic specific antibodies that neutralize pathogens and facilitate opsonization or activate complement mediated destruction. While memory B cells persist for years to rapidly respond to subsequent exposure to the same antigen. In T cells, activated cytotoxic T lymphocytes directly eliminate infected or aberrant host cells, and memory T cells remain quiescent yet highly responsive, enabling faster and more robust secondary immune response. During lymphocyte development, a crucial selection process known as negative selection eliminates cells that strongly recognize self antigens, thereby preventing autoimmunity and maintaining tolerance to the body's own tissue. Through this precise multi step mechanism, the clonal selection theory elegantly accounts for the four cardinal features of adaptive immunity. Specificity, by activating only lymphocytes whose receptor match the antigen. Diversity, through generations of a vast repertoire of unique lymphocytes. Memory, via persist for long lived memory cells that accelerate secondary response. And the ability to distinguish self from non self, ensuring that immune responses target only foreign antigens. Let's take a look at the diagram. So, clonal selection begins when a specific B lymphocytes, one of many circulating in the body, encounters and binds to a foreign antigen that precisely matches its membrane bound receptor. This antigen, which may originate from a virus, bacteria, or other pathogen, activates the B cell, which then becomes the antigen binding lymphocytes. Following activation, the B cell undergoes mitosis, producing a clone of genetically identical cells known as B lymphoblasts, a process called clonal expansion. These lymphoblasts then differentiate into two types of cells: plasma cells and memory cells. Plasma cells function as effector cells and secrete large quantities of antibodies that are specific to the original antigens. These antibodies bind to the antigen to neutralize it or mark it for destruction by other components of the immune system. At the same time, memory B cells are formed and remain in the body for extended periods. They retain an specificity for the original antigen and allow for a faster and stronger immune response upon further exposure to the same pathogen, providing long term immunity. Through this process, the immune system effectively eliminates the current infection and prepares for the potential reinfection. Now, adaptive immunity exhibits four fundamental features, all of which are elegantly explained by clonal selection theory. Specificity refers to the precise ability of each lymphocyte to recognize a particular antigen. Even B cell expresses a unique B cell receptor, and every T cell expresses a unique T cell receptor, each capable of binding only a single antigenic epitope. When an antigen enters the body, only the lymphocytes whose receptor perfectly complement that antigen are selected and activated. This ensures that the immune response is highly targeted, attacking the invading pathogen while leaving unrelated antigens untouched. Diversity. Diversity is the immune system's capacity to recognize an immense array of antigens. Through processes such as genetic recombination, lymphocytes generate a vast repertoire of receptors, each different from the others. This diversity allows the immune system to respond to virtually any pathogen the body might encounter, including previously unknown or rapidly evolved organisms. Memory. Memory is the ability of the immune system to remember past encounters with specific antigens. When a lymphocyte is activated, some of its progeny differentiate into long-lived memory cells. These cells persist in the circulation for extended periods, often for years, and upon re-exposure to the same antigen, they respond more rapidly and robustly than during the primary response. This mechanism underlies that enhanced secondary immune response and forms the scientific basis for vaccination. Finally, the immune system's ability to distinguish self from non-self. It ensures that it targets only for an antigen while avoiding damage to the body's own tissue. During lymphocyte development, immature lymphocytes undergo a process called negative selection, where cells that are strongly recognize self antigens are either eliminated through clonal detection or deletion or renders non-functional through energy. Only lymphocytes that recognize foreign antigens are permitted to survive and participate in clonal expansion. This self-tolerance prevents autoimmune reactions and ensures that the adaptive immune response is both precise and safe for the host. That's all for this video. Thank you.